Alright, so we're going to pick up now with our third section in the membrane and cell process chapter, and we're now looking at active transport. So, in the last section, we talked about passive transport being a way for cells to get things into themselves or out of themselves free of charge when it comes to energy, so it doesn't cost them any ATP to move things in or out using passive transport. But the caveat there is that for things to move through passive transport, those things have to go with a concentration gradient. Down a concentration gradient means the same thing. They have to go from high concentration to low concentration. So active transport is going to be the opposite of all that. Active transport is going to move things from low to high concentration. We can say things are moving against their concentration gradient as such. And there's going to have to be some ATP spent. Now, in addition to that, we're going to be talking about using some proteins that are involved in moving things with active transport. And typically, these carrier proteins are called pumps. So a pump is going to be a transport protein that is geared toward moving things against their concentration gradient with the use of ATP powering that pump. And so the ATP, of course, is coming from cellular respiration. We're going to be talking about that in an upcoming chapter as far as how that ATP gets made. But for right now, we're just saying it's ATP, right? ATP is the main energy currency of the cell, and among other things, ATP can be used to power active transport. So a good example of active transport is the sodium-potassium pump that we find in neurons, right? So nerve cells. And so it turns out that without getting into, why this needs to take place so much, nerve cells need to be able to maintain what's called a resting potential. And resting potential, in large part, is maintained by the sodium-potassium pump. So to kind of set the stage for you, a nerve cell at rest is going to have a relatively high concentration of potassium ions inside the cell and a relatively high concentration of sodium ions outside the cell. Right? So high potassium in, high sodium outside. Now this is a pump, so we know we're going to be using ATP, and we know we're going to be using this pump to move both of these two ions against their respective concentration gradients. So if we know that potassium ions are higher inside than outside, we know we must be moving potassium ions into the cell because we want to go from low concentration to high concentration. Same thing for sodium. Sodium, we know we're going to be kicking out of the cell from low concentration toward higher concentration because there's more sodium present outside the cell than there is inside the cell. And so without delving too much deeper into this, essentially one cycle of the sodium potassium pump is going to involve the removal of 3 sodium ions from the cell in return for 2 potassium ions being moved back into the cell. Right? So again, high potassium inside, high sodium outside, 3 sodiums are kicked out, 2 potassiums are brought back in. Essentially, every time the sodium potassium pump cycles, we're going to be losing a negative 1 charge inside this cell because 3 pluses are leaving and only 2 are being brought back in, so the cell's charge is going down every time the sodium potassium pump cycles, essentially, and every time ATP is spent in the process. Now, I don't want to go any further than that right now, but if you take anatomy and physiology, if you take principles of biology 2, we get into more of, what else helps that negative charge kind of build up, why that negative charge is important, and what happens when a neuron actually fires and the inside of the cell becomes positively charged as opposed to negatively charged. But that's all later coursework if you decide to take more biology courses, if I don't scare you off with this one. All right, so this is a calcium pump in this picture. And so in this case, we have low calcium inside the cell, high calcium outside the cell, and here is our calcium pump. So we're actually going to bond calcium ions to this pump. We're also going to bond ATP. So ATP gets broken down, calcium is kicked out of the cell, and ADP and phosphate are released. And once ADP and phosphate are released, the pump goes back to its original configuration. It can pick up another calcium, and as long as we have more energy, that next calcium can also be moved out of the cell. All right, so the breakdown of ATP is going to yield ADP and phosphate. Adenosine triphosphate breaks down to adenosine diphosphate and an extra phosphate group. All right, so that's a calcium pump. So to kind of summarize so far, we've looked at simple diffusion, where we have particles going across the membrane without any help whatsoever, no protein help at all. They're going right through the phospholipid bilayer. Of course, they're going with the concentration gradient, and these particles are the ones that are hydrophobic because, of course, they have to travel across that mostly hydrophobic interior part of that membrane. We also dealt with our facilitated diffusion, where we had our channel proteins, and in this case, in this picture, a carrier protein that's moving things, again, with the concentration gradient across the phospholipid bilayer from high concentration to low concentration. But in this case, we have a transport protein or a carrier that's going to help to move this glucose, for example, across this membrane. And then with active transport, now we're moving things against the concentration gradient, so now We're going from low concentration to high concentration, and not only is there a protein involved, there's also energy expenditure involved as well to move those molecules, in this case, into the cell. Now, the last type of transport that we're going to talk about, we've done passive, we've done active. Now we're going to look at what's called bulk transport. And the reason why it's called bulk transport is because we're going to be moving some really, really large molecules either into a cell or out of a cell. And these things are way too big to fit through any carrier, any channel, or especially any phospholipid bilayer without any help. So we have to find other ways to move these things into or out of cells. Typically, that's going to involve the use of vesicles, right? So transport vesicles, like we talked about back in our previous chapter, are little bubbles of membrane that can be used to move things inside of a cell, and in this case, move them completely out of the cell or into the cell. So transport vesicles are going to be kind of the keystone of both of the two main types of bulk transport processes. And these two processes are exocytosis and endocytosis. Exocytosis, exo is out, so exo is like exporting things out of a cell, and endocytosis means bringing things into a cell, again using vesicles to capture those particles. We're going to look at a couple different types of endocytosis. Really, exocytosis is just one main thing, but there are some subtypes of endocytosis that we'll look at as well. So with endocytosis, essentially what's going to happen is that a cell is going to use a little bit of its plasma membrane to surround and capture some kind of large particle outside the cell and bring it into the cell inside of what's called a vesicle. Right? So essentially, whenever a cell does endocytosis, it actually ends up having just a tiny little bit less plasma membrane than before it did the endocytosis. There are, like I said before, a couple different subtypes. We have phagocytosis, which is where cells take in solid particles, literally means cell eating. We have pinocytosis, where cells take in large amounts of liquids. So pinocytosis is literally cell drinking. And then we have a third type called receptor-mediated endocytosis, where cells capture very, very specific particles, and only when the receptors for those particles have been fully sort of saturated by those particles do they actually get brought into the cell. So that one's a very specific subtype that we'll take a look at, like one example of that a bit later on, and we'll see kind of where that can become important. Exocytosis, not really any subtypes. It's just getting stuff out of the cell, large molecules being exported from the cell. In this case, a vesicle containing some kind of large molecule comes off of, say, the Golgi, for example, travels to the membrane, fuses with the membrane. So the membrane, the plasma membrane actually gets a little bit bigger, and that particle is then exported out of the cell. All right, so for things like, for example, endocrine glands, cells that are producing large proteins of materials way too big to fit through any carrier or channel protein, they can be exported from those glandular cells through the process of exocytosis. Bulk transport is very, very expensive. Cells don't do it unless they absolutely have to. It really has to be worth it because it burns up a bunch of ATP whenever they do it. But, if you're dealing with cells releasing hormones, that's a pretty important thing. That's probably going to be worth it. If you're looking at exocrine glands like tear ducts or salivary glands or pancreatic cells that are releasing things into the intestine, all of those things are released through exocytosis, like we just talked about. All right, so the idea of receptor-mediated endocytosis, the one that's probably the most complicated of all the subtypes. Imagine this being a cell, and this, of course, is the plasma membrane of the cell. There are all these little receptors outside this cell. And so whenever a cluster of receptors grabs the molecule that they're geared to fit, they form what's called a coated pit. So now we have our coated pit forming. We have all the receptors bound to all these little green particles. And eventually that coated pit becomes a transport vesicle where the receptors and the molecules they've captured are brought into the cell, and they can then be processed. All right, so this is actually really important in our body when we're trying to get cholesterol out of our blood. So after a cholesterol-rich meal, you don't want to leave that molecule in the blood, because if you do, it's going to start to be deposited in places in your body where you don't want it, like in coronary arteries or blood vessels in your brain or all kinds of places where you really don't want cholesterol deposits. It's kind of important to get that stuff out of the blood and into certain cells in your body. So those cells that are geared for taking up cholesterol are going to capture those molecules and bring them into themselves using receptor-mediated endocytosis. Now, one thing that's kind of interesting is these receptors are almost always at least partially protein-based, if not completely protein-based. And as with any protein, we have genes in our genome that tell our cells how to build these receptors. If somebody is missing the gene, or if somebody has a gene to make an improperly formed receptor, then they may not be able to bring in that molecule into their cells anymore. And so we'll see an example of that here in a few minutes, of a situation where somebody who has inherited two faulty copies of the gene they need to make these receptors actually have chronically high cholesterol and very frequently die at a relatively young age because cholesterol, like I said before, stays in the blood and ends up being deposited in places where they don't really want it to be. So a bunch of pictures now. This is pinocytosis. So we have a cell capturing large amounts of liquids being brought into the cell, type of endocytosis. We have an actual photomicrograph here. Actually, this is an electron micrograph here of cell doing pinocytosis where it's capturing fluid from the environment. This is the cell. This is the environment. These are all vesicles forming with fluids inside of them being brought into the cell. Kind of the same thing here, but now it's stained, so you can actually see extracellular environment, plasma membrane, fluid being captured. This is the rest of the cytoplasm inside the cell here. This is a macrophage. This kind of whitish cell is a macrophage, which literally means like big eater. These are one of our types of immune cells that are used to, they're kind of around all the time in the body. They're not really something that is very specific to certain kinds of pathogens, for example, that might be in your body, but they're kind of there all the time, and they're looking for anything that might get into your body that shouldn't be there. Macrophages are going to go along, they're going to take those out by consuming them, by phagocytizing them, and then destroying them accordingly. In this case, a macrophage has found a yeast cell, which is about to be destroyed by the macrophage once it's done phagocytizing it. This is a white blood cell of a different type, consuming bacteria. So this maybe is a neutrophil, for example, taking out bacteria, phagocytizing them, destroying them, getting them out of the system so they don't have a chance to cause illness. This one, amoeba, is a protist. Paramecium is also a protist, but this amoeba is surrounding the paramecium. It is going to phagocytize it. This amoeba would then have vesicles inside of it, lysosomes, for example, that might be able to digest that paramecium once it's consumed by the amoeba. So this is like predator-prey, but on a microscopic scale. The predator is this thing surrounding the prey. Through phagocytosis, it will eventually destroy it. Again, receptor-mediated endocytosis, again, we've got those receptors, we've got the molecules that are being captured, coated pits forming, eventually vesicles bringing those molecules into the cell where they can be dealt with accordingly. And then the genetic condition I talked about before is called familial hypercholesterolemia. All right, so you don't necessarily need to know that name. It's kind of a mouthful, but familial hypercholesterolemia is a situation where somebody inherits two genes, two versions of a gene, one from each parent, right, that end up producing a faulty protein receptor on the outside of the cell. So even though they've got receptors for cholesterol, if the receptor is the wrong shape, it can't capture the cholesterol, cannot bring it into the cell, and so the cholesterol stays in the blood where, like we said before, you really don't want it to be. And so at that point, you start seeing higher incidences of heart disease and possible stroke and blood pressure issues and all kinds of things that result from abnormally high cholesterol in the blood. These are folks who eat just the healthiest diet in the world, and they run 10 miles a day, and they die of a heart attack when they're 25 because their cholesterol levels are through the roof, right? So it's a genetically high predisposition toward elevated cholesterol. More exocytosis, so we've got particles that have been produced by the Golgi, the vesicles fusing with the membrane. Membrane gets a little bit bigger, right? So exocytosis is membrane gained. So the vesicle becomes part of the membrane, and the particles are then released into the environment, where they can be perhaps entering the blood, maybe they can be used by some other cell nearby, but they've been released from that cell. Same idea with exocytosis there, and a couple of actual micrographs of exocytosis from a cell. This vesicle is fused with the membrane, and these molecules are being released out into the environment. So this is endocytosis, right? And that is this is pinocytosis down here. So phagocytosis, pinocytosis here. Both are types of endocytosis. The difference is what's being consumed. Is it solid matter? Is it liquid? That's kind of the distinction between phago and pinocytosis. So the very last section in this chapter is to look at different types of cell-to-cell connections. And so there are three. The first type of cell-to-cell connection is called a desmosome, or what's sometimes called an anchoring junction. And so desmosomes, or anchoring junctions, are found in places in the body where cells are subjected to sort of like high stress type situations, large amounts of physical stress, things that might tend to pull cells apart from each other. So places like in your skin, for example, places like junctions between muscle cells where there's a lot of physical force that might threaten to pull the cells apart from one another, tear tissue, and so forth. We have desmosomes there to help provide a firm, steady anchorage point there for those cells so they can't be ripped apart quite so easily. Second type of connection is called a tight junction. This is a relatively straightforward one. This is going to be found in parts of the body where waterproofing is necessary. So places where you want to make leak-proof connections between the cells would tend to have tight junctions. The cells in this case are pressed together with proteins, as opposed in the case of the desmosomes. In this case, the proteins actually go from the cytoskeleton of this cell through its membrane into the extracellular environment, and then into more desmosomes in the next cell, which then connect to that next cell cytoskeleton. So these two cells are like riveted together. Over here, these cells are pressed together with proteins that make them waterproof. So a good example of this, places like the urinary bladder, places like the small and large intestine, are held together by tight junctions because you really don't want materials leaking out of those respective organs. We want the cells to be pressed together and very much waterproof as a result. Third type of connection, there's actually two subtypes depending on if we're dealing with animal cells or plant cells. In animals, we have what are called gap junctions. Gap junctions are places like in cardiac muscle, where there are cell-to-cell connections that allow things to be passed back and forth very quickly among the different cells. So kind of the idea down here, you see it, we've got two plasma membranes from two separate cells, and there are gap junctions between these two cells, like a little tunnel that can allow matter or electrical impulses to travel through them very quickly without actually having to physically cross this membrane. They go from cell to cell to cell to cell very, very fast through these gap junctions. Really important in cardiac muscle. You really want all those cardiac cells to contract at the same time without any sort of delay between them, so your heart actually functions as a single physical pump or unit, as opposed to just a bunch of cells contracting whenever they feel like it. In plants, we call these plasmodesmata. Kind of the same idea. A lot of times in plants, we're dealing with water being transported from cell to cell. Same idea as a gap junction, different name, but we've got water being moved from one cell to the next, to the next, to the next very quickly because they're essentially connected through these plasmodesmata. So with that, we're going to wrap up this chapter. The next chapter that we're going to see is going to deal with energy and thermodynamics and enzymes especially. So we're going to wrap this one up now, get it posted, and we'll start up on our next chapter.